In this lecture, we will continue our discussion of methods in neuroendocrinology. The lecture that corresponds to this one is the Methods in Neuroendocrinology 1C. We'll discuss primarily neurochemistry today and follow it with a couple of abstracts from research articles that demonstrate some of the utility of these methods in neurochemistry for at least related to neuroendocrinology, if not a bit remote from that. So let's go over to the computer and begin this discussion. As I said, it's a variety of methods in neurochemistry that we're going to evaluate. The first is in vivo microdialysis, the second is in vivo microvoltometry, and the third is push-pull perfusion. So we've talked about neurotransmitters and neuropeptides and release at synapses, producing synaptic concentrations. Even explain the idea of the ability of these chemical messengers in the brain to escape from synapses and get into the interstitial space and act as neuromodulators, that is, not transsynaptically, so extrasynaptically. These methods are available to sample that interstitial fluid. We can, of course, get into an individual synapse, but we can lower some type of a device into the brain parenchyma, the brain tissue, and evaluate what is in the interstitial fluid compartment, and we'll explain how each of these different methods works. Before I begin to talk about that, we need to first understand the reason for this term in vivo, and that is, of course, chronological or historical in nature. Originally, even though biochemical methods existed for detecting and quantitating many of the classical neurotransmitters, such as the monoamines, in homogenized samples, it was all performed ex vivo. You had to sample the tissue, destroy the tissue, obviously, so there's euthanization or surgical removal and destruction of the tissue to evaluate that content. And the term content is really important. With those ex vivo methods, you're homogenizing both the axons, if you want to think of a neurotransmitter at an axon terminal site, you're homogenizing the intracellular sample as well as the extracellular. So when you get a content through that method, through a microdissection type of a method as that, you're evaluating both intracellular and extracellular concentrations for the content. In vivo methods sample only the extracellular or interstitial fluid compartment and thereby allow the more direct evaluation of synaptic levels that might reflect changes in release or changes in reuptake or changes in catabolism or even synthesis. So all those approaches we talked about for increasing or decreasing synaptic concentrations of neurotransmitters can be reflected via utilization of these in vivo methods. And we'll differentiate between them as we proceed. So let's start first with the in vivo microdialysis. Here on the right-hand side we see a dialysis tube, so it's a dialysis membrane. I'm just assuming that if you're this advanced in biology, you've used a dialysis membrane somewhere along the way to dialyze molecules. And we know that these dialysis membranes have different pore sizes, either allowing only water to escape or water in small solutes, or larger molecules such as the neurotransmitters, and then even larger molecules, if you have a high porosity, to have peptides pass through it. But large proteins and particulate matter obviously will not pass through it. So we have this dialysis membrane as part of a tube, and we see that there is a flow in and a flow out This is called the dialysate. So the dialysate in, where the affluent, AFFLUENT, is going in, just like afferent was for sensory information coming into the nervous system as we studied, and the effluent, EFFLUENT, is the dialysate coming out of the dialysis tube or apparatus. So the effluent would carry the molecules of interest that we would want to be drawing from the interstitium of the brain. So on the left-hand side here is a crude illustration of the brain parenchyma. We see a synaptic terminal releasing dopamine into the synapse, creating a synaptic concentration of about 0.5 molar, and that can diffuse away, giving about maybe a 5 nanomolar concentration available to be picked up into your effluent dialysate, excuse me. And then the escape through the interstitial fluid compartment that might be picked up with the second method we'll talk about, the voltammetric carbon fiber technique for dopamine of about 0.1 to, got to look close at that, 2 micromolar. So you can see that the distance away from the collective synapses for the transmitter of interest can be reflected by the concentration detected or produced through various methods. So you can see that the microdialysis already is an ex vivo analysis of the analyte or analytes, A-N-A-L-Y-T-E, or S for plural, whereas obviously here they're talking about putting a carbon fiber for voltammetry right into the brain, so it is completely in vivo. You don't suck any juice or content from the brain. It all stays right there. Obviously, we are lowering things into the brain, so we are producing a localized damage to the tissue and disruption of some of the capillaries and vasculature in the area. So it's not a perfect method, but it is still more reflective of synaptic concentrations and all the dynamics that contribute to that than any ex vivo post-mortem method. On the right-hand side, there are 3 physical processes that are being represented. The first is diffusion with the parameters that are important for that, and we're not going to focus on any of those parameters. Ultrafiltration and osmosis. So all 3 of these physical parameters contribute to the movement across the membrane in both directions, and obviously it depends upon the affluent solution that you are feeding into the dialysate tube as to what will come from the interstitial fluid into there or go from the dialysate into the brain interstitial fluid. So there's a lot of detail that we're going to pass by. Overlook, I think, is the term I'm looking for there. And primarily for our interest at the moment, this would be an affluent or a dialysate coming into the dialysis tube designed to allow the osmosis of many molecules smaller than or equivalent to the classical neurotransmitters, which is the focus of our attention at the moment, or to include peptides if the pore size and these dynamics changed a bit. But keep in mind that you can also add molecules to the dialysate flowing in, the affluent dialysate, and if it is a molecule that doesn't exist in the interstitial fluid compartment of the target tissue, the brain tissue in this case, you can deliver that molecule into the brain tissue. For example, we are using it to deliver amphetamine into rats' reward centers when they press a lever in one of our experiments at this time. In the lower panel, we can see that the microdialysis approach can be used for many different applications. We'll see another example of this in a moment. Anywhere that you have a blood capillary with flow in and flow out, all these same dynamics apply, physical properties. You can drop in a microdialysis catheter or apparatus to accomplish the same thing, to sample the extracellular fluid or to contribute something to the extracellular fluid to evaluate the biochemistry of the region, including the cells, at least as far as their interface with the extracellular fluid compartment. In the left-hand panels, what we see in the upper part are 2 rats on running wheels that are fitted with a chronically implanted microdialysis cannula, and the tubing comes up through here, and we'll look at the details in a moment with reference to this. Here is another one placing the rat in a bowl. So obviously, if you have rats with a chronically implanted cannula somewhere in their brain and tubes that come in and out, you can't just let them run freely out in the field. There has to be some limitation as to their freedom to roam around, but certainly in their home cage or a running wheel, as we saw, or in that bowl, then you can perform the experiments. This creates an odd type of an environment, especially if it isn't their home cage, and it can potentially contribute to the data that you receive, but obviously you design the experiment to take care of that, to control for all those parameters. So in this image on the right-hand side, we see again the basic type of a dialysis tube. It's solid at the end and then connected to additional tubing up above, as you can see here, and then the dialysis membrane is a cylindrical structure down here at the proximal end that would not be implanted. Then you have the inflow and the outflow tubes attached, and it can either be a common chamber in there, or the inflow can be all the way to the bottom. You can see that this is a metal cannula coming all the way down to the bottom with an opening there to push the fluid out, and then the dialyzing fluid, I'm sorry, I have it reversed, coming in around the outside and then pushed into the inner cannula to come out. Sorry about that, I should have followed my arrows. Anyway, we've got fluid coming in and out across the dialyzing membrane, and we have the dynamics bidirectional based upon all the physical properties that were mentioned before. You can see a subcutaneous catheter is going to have a much longer dialyzing tube length and much shorter for the brain. Now we're beginning to see many of the limitations of this method. First is you might be sampling from a tremendously large area of the brain if you're interested in just, say for example, one little part like the paraventricular nucleus of the hypothalamus to study neurotransmitter release during stress or something. So there are artifacts, there are confounding, contributing factors to this study. It is becoming more and more refined all the time, but still it has limitations. The other limitation is you have to collect samples. The minimum I've seen is 5 or 10 minutes, which relative to dynamic synaptic events is a very long period of time. So you're looking at a 5 or 10 minute sample, a certain concentration of your neurotransmitter drawn from the extracellular fluid compartment of your area of interest, and you're trying to relate that to synaptic events that may have occurred in millisecond time frames. So there is a temporal resolution limitation to this method, and we'll see more of that as we proceed. So here is an illustration, the type of which you might see on an exam, where you would have to match the labels to the structures, the items included in this apparatus. So this is the illustration. Here we see the actual picture. This is the one for a rat with that bowl. The pumps are shown here, feeding in, and then the effluent comes up through this apparatus into an auto sampler to collect the fractions of the samples, say 5-minute fractions, 10-minute fractions. Most you will see will be 30-minute or 1-hour collection periods to get a large enough volume in each fraction in order to process it through the separation method and the detector in order to get a signal. So let's take a look at this illustration here. This is one designed for bedside human microdialysis, such as subcutaneous or intravenous. Obviously, there has to be the pump to push the fluid in, and then B, we see the the catheter, and then C, up here, we see the collection in microvials, and then the microvials brought to the bedside microdialysis analyzer. D, I guess it went off the screen there, but it is labeled D up here. And then E, labeled here up above, is the comparable, trend curves that are displayed over a period of time. So the D, the processor, the analyzer, has to separate and analyze your different chemicals of interest to give you the signal to interpret. So it'd be comparable to this display for an echocardiogram or blood pressure or intracerebral pressure as seen commonly in intensive care units. So someone obviously was trying to design this to market in the clinical setting. But it was convenient to illustrate the pieces and parts that would be needed for any such study. Let's take a look at this image of an analyzer, pieces and parts of which you might need to label on an exam as well. This is a high-pressure liquid chromatography instrument, HPLC. The P is also used for performance, high performance, liquid chromatography, so it's liquid. So we have two injectors with the pumps attached to this system. So two samples can be run simultaneously. They are fed through this pre-pump to feed the samples through this filtration column that will clean out any dirt. Then it will pass through the columns. Here we see the two columns. So we can inject two at once, and then these pumps will pump each of those through this system and through the separate columns. So it's a rather sophisticated system. Cost you several tens of thousands of dollars. After the separation in the columns, we'll talk about the types of columns in a moment. And obviously you have liquid flowing through here referred to as the mobile phase. So somewhere out here there's got to be a bottle of liquid feeding into this pump system where you can inject. And the pump takes that mobile phase, runs it through this additional pump to pre-filter, and then runs through the fixed phase, which is within these columns. And we'll talk about the nature of the fixed column, the fixed phase within the columns, to separate before detection different types of molecules. So the fixed phase is in the columns, and then after the columns you get the separation of your molecules based upon the nature of the column, and then within here there's a detector, and the detector will either be electrochemical, so we'll look at an abstract where they use HPLC EC, electrochemical detection. Can be ultraviolet for peptides, and depending upon the procedure, might be a couple of other types of detectors, but ultraviolet and electrochemical are the two most common ones. And then obviously this would connect to a computer with software that would give you your signals in a graphic type of display, allowing you to quantitate. So we'll take a look at that as we proceed. Could also feed out to a paper printer on graphical paper or chart paper to give you results that way. That's kind of the old way. We don't see that too commonly anymore. Now let's consider the type of column that might be used for the fixed phase through which the mobile phase in your sample would pass. We can see on this graph, on the y-axis is pore size, and on the x-axis is the polarity of the packing materials. So it's either going to be largely polar over here on the left, or small polarity on the right hand. On the right hand side. The large polarity and the larger pore sizes would be used for sugars. Vitamins fall somewhere in here on the polarity. We can see it circled there. The polycyclic aromatic compounds fall in this area somewhere. Low molecular weight substances, medicines, organic acids, and food additives, etc., fall into this arena here. We see this one type, the C8. This is the most common used for the neurotransmitters, monoamines, that we'd be considering. And the peptides and proteins fall out into this small polarity level and range in the higher to the highest pore size that would be required. So obviously, the smaller your molecules, the smaller the pore you want in your fixed column, and depending upon the chemical nature of the molecules, you want it either more polar or less polar, so that there's both a molecular sieve based upon size that's created, so the smaller molecules will slip through faster, and those molecules that have the chemical characteristics that are not resisted or retained by the chemical nature, the polarity of the column, will go through fastest. It'll be the first ones to show up on your software screen with a blip on the screen, and then the larger molecules with the higher polarity, in the case of the neurotransmitters, where you have the lower polarity, where we have a small polarity or moderate level of polarity in the packing material. Those will be retained longer and will come off later. So it's a typical type of chemical separation based upon the size and the biochemical properties of the molecules. And it is under a high pressure with the mobile phase and the liquid phase, both of which have to be designed to create a biochemical situation for your molecules so that they separate nicely on the type of fixed phase column. So the molecules that you inject will take a certain period of time to get through the columns and reach the detector. That's called their retention time. So each molecule is going to have a different retention time depending upon the mobile phase and the fixed phase that would be used. So in the case of the in vivo microdialysis, those samples would be collected as we saw, and then each sample injected into such an HPLC system as we had observed. Electrochemical detection is the most commonly used for the monoamines, and this illustration is showing trace enrichment, LC. They don't have the HP on here, but it is HPLC, liquid chromatography, EC, electrochemical detection for the catecholamines. They used an enrichment column, cation exchange, and the analytical column was octadecyl bonded, so that was the C8, 8 carbon type of bonded silica column, the fixed phase, with a 5 micrometer pore. The counter ion was potassium, the matrix was brain tissue, so this is ex vivo. We'll look at the figure legend in a moment. There was an offline purification. They used aluminum oxide to precipitate out the catecholamines and basically to extract and concentrate the catecholamines before injecting them. Let's keep that PIP off so we can see all of the traces. So here in this lower portion of the figure, we see the chromatograms of a calibration curve point and extracts of micropunch dissected medial and central amygdaloid nuclei generated by online trace enrichment HPLC-EC assays, the effects of tyrosine hydroxylase inhibition with alpha-methylparatyrosine in this image here. So we can measure the decay in the signal and get a feeling for a half-life. And an anesthetic, gamma-butyrolactone, GBL, are illustrated. The anesthetic should inhibit release, so we can get an assessment of release rate under the conditions. Now again, this is ex vivo, so they had to administer these drugs to animals, wait a period of time, euthanize the animal, dissect the tissue, microdissect the tissue, homogenize the tissue, and then go on. So these signals reflect total content, intracellular and extracellular, in these samples. If this was from microdialysis samples from these animals, then these would be extracellular only. The interstitial concentrations would be reflected. So let's take a look first at the standard. It's a 1 nanogram standard that was extracted with the aluminum oxide, so they included dopamine and norepinephrine. On the x-axis we see time, so retention time differs. So this is the first peak that we saw, and then this is the later peak. So dopamine comes off early, it has a shorter retention time than does the norepinephrine, and this is probably just some kind of dirt out here in the later part. We see this DHBA. This is a common internal standard. The y-axis is volts, is showing current. Here we see 0.2 nanoamps on this scale bar. So you can quantitate, and if you know your concentration, then you can convert nanoamps into the nanogram concentration. So this is the standard. 1 nanogram of dopamine produces this many nanoamps. 1 nanogram of norepinephrine produces this many. The DHBA is an internal standard that persists in our tissue samples. So here's the DHBA in each of these. It's this second peak. And here we see the medial amygdaloid nucleus. The dopamine is a very small peak. We can hardly see it here. It's not even 0.1 nanoamps. So very little dopamine contained within the medial amygdaloid nucleus. Here's the DHBA and the norepinephrine out here at the later about 10 minutes. This is a 5-minute scale, so I'm assuming about 5 minutes to dopamine and 5 minutes additional to get the norepinephrine under these particular mobile phase and fixed phase conditions. Now in the central amygdaloid nucleus we see the control, the alpha-methylparatyrosine to inhibit the tyrosine hydroxylase, and the anesthetic, the gamma-butyrolactone. And we're looking at the, comparing to control, look at the tremendously high signal for dopamine, and norepinephrine is a little bit higher than it was in the medial amygdaloid nucleus, but not as high as what the 1 nanogram produced in the standard. So the DHBA isn't quite as high as it is for the sample processed from the medial amygdaloid nucleus, but the reason that it's an internal standard is you get a ratio of the signal in nanoamps to the internal standard, and that normalizes your data. So the difference between the internal standard here and in the control is very little, and if the proportion drop off between the medial amygdala and the central amygdaloid nucleus is large enough, then this signal might even be equivalent to the 1 nanogram. However, the proportion drop off here doesn't appear to be quite so great as to bring our dopamine signal down that much. It's still a very large peak for dopamine, and we can see the norepinephrine here. After the alpha-methylparatyrosine, we don't know how long after, but at some time point after the alpha-methylparatyrosine, inhibition of tyrosine hydroxylase, we see that the dopamine signal decreased significantly, whereas the internal standard peak did not change, and the norepinephrine decreased just a little bit as well. If we block release with this anesthetic, then we see that dopamine peak is decreased as well, but the oddly enough, the norepinephrine increased. Excuse me. So this is a bit odd for the norepinephrine, but it means that there is a pretty high rate of dopamine release. It might be a third of the total content is due to release over that period of time, and looks like nearly half of the dopamine is due to new synthesis over that period of time. So this is a way that we could interpret the data in this case. So now let's take a look at a figure from a publication as an example. This is from 2001 publication in the European Journal of Pharmacology, volume 411, issue 1-2, pages 115 through 122. And the title is The Functional Effects of Corticosterone on 5-HT1A and 1B Receptor Activity in the Rat Brain in vivo microdialysis studies. So the graph we see in the center here on the x-axis is the fraction, and we'll take a look at these in a bit more detail in a moment. These, I believe, represent 30-minute samples, so each fraction is a 30-minute sample. So you can see fraction 1, 2, 3 lines up, 4, 5, 6 lines up. So each of these are 30-minute fractions, and then that fraction is run through an HPLC to detect serotonin, and it is reported as the percent of the basal level. So here we see before treatment with this GR127835, which is a 5-HT1A and 1B antagonist. These are the presynaptic receptors, the autoinhibitory receptors. So if you have an antagonist to them, then release should increase, and the extracellular, the synaptic concentration, and thereby extracellular concentrations of serotonin should increase also. So here you can see the time point, or the fraction point at which this drug was injected into two sets of animals. The triangles show the ones that were co-treated with corticosterone. The circles show the data points for those animals that were treated with the vehicle, whatever was used to dissolve the corticosterone. So you can see that by fraction 5, the extracellular serotonin levels begin to increase. This is 100 to 150 here. So this is about 25% above baseline, whereas the corticosterone treated animals, it's roughly 160. So it's about 60%, so twofold, a little bit more than twofold, greater than the vehicle-treated animals. And this separation continues throughout. We can see for each data point from each fraction, there's the standard error of the mean. The point is the mean plus or minus the standard error of the mean. We can see here they begin showing significant differences, at least at these 2 points, when compared to the corresponding fraction on the vehicle-treated animals. So the analysis on this, because there are two drugs and two different levels. No, excuse me, there's just one drug with two different levels for that drug. This is administered to all animals, so it isn't a factor. So this is a one-factor, one-way analysis of variance with repeated measures that was performed on this. That must have been significant. And then these data points with the single asterisk were significant at the p less than 0.05 level when using the Newman-Cool's post hoc test. So if you don't understand those statistics, you may want to take a course in statistics, or I also teach the Research Ethics and Responsible Conduct of Research course, where we cover that information at an introductory level, at least, to assist you with your understanding. Now we can begin to talk about in vivo microvoltammetry. So with this method, there is a probe that is designed that can be chronically implanted, and at the tip of that probe is an exposed surface of material that is a dried paste of a variety of different compositions that give it different properties. And basically what is happening is you have a certain distance of that flat surface that is exposed, and the chemicals that find their way somehow, we know through Brownian motion and diffusion, into this area in the interstitial fluid compartment can interact with that electrode surface. And we'll take a look at the graph here to appreciate that first before we just talk about the instrumentation that would be required. So with the in vivo microvoltammetry, obviously it's micro, that means it's micro scale, so this distance is small. These probes should be small if you're going to implant them in the brain. They can be larger if you're doing a solution detection method, but we're more interested in the in vivo, so it would have to be small to be implanted into the brain. The way that this is accomplished, we see in this figure the current in microamps versus the potential. So a potential is applied to the paste, that dried paste, and it is changed. Here we see from a minus 0.2 to a plus 1 potential and voltage that is applied to that. And the molecules that are nearby are going to undergo an oxidation reduction reaction and will generate a current, in this case microamps. We'll see in the brain. With in vivo, it's more like a nanoamp type. We're not going to try to become experts in any of these methods, as we always remind ourselves as we go through these lectures, but we do want to have some appreciation for how they are accomplished, so we won't get into the details of each of the different methods and how the voltage is applied and what the current means. But anyway, you can quantitate the current. You can see a signal, you get a peak, and that gives you something that you can measure. So instrumentation is required for this. It's a bit sophisticated because you have to have a temporal application of the voltage and be able to concurrently measure the current generated at the disk. There has to be some time between sampling so that you can allow for the redox reactions to reverse themselves in this area, or you can reverse them by sending a current through it, changing the voltage potential again to try to change the chemicals back to their native status. So the signal comes in, you've got all the controllers, and then the signal comes in, feeds into software in a computer, and then you generate your signal, similar to the chromatography that we'd seen before. So there's these types of software packages where you set all the parameters, the sweep times, and all these things. And the purpose of an experiment including this method might be to compare and contrast three different voltammetric methods by using each method in turn to examine the very same analyte solution. And the methods studied are the cyclic voltammetry, related disk voltammetry, and chronal amperometry, which is the one that I'll focus on for a moment. And then each method is used to determine the diffusion coefficient of the analyte. So this involves, to have a truly detailed appreciation for what's going on, your physics and biochemistry has to be very strong, and your chemistry in general. So one type of approach is cyclic voltammetry. There's a sweep method, as we saw, where the voltage is swept in both directions during a drop, typically less than 1 second. The type of electrode is a DME, glassy carbon, or platinum electrodes may be used. The method looks at oxidation reduction and if the reaction is reversible, and it's used to study the redox in organics and organometallics. The differential pulsed polarography is one where the potential is applied as a ramped square wave, as we see here, and the current is sampled just before and near the end of each pulse so that you get peaks at different voltages applied and different signals. So let's take this to an example from a paper, actually on which I'm a co-author, where we used semi-differential microvoltammetry to produce these microvoltammograms. So you can see on the x-axis of each of these figures in the upper panel is the applied voltage. So from about the negative 0.1 out to positive 0.4. And on the y-axis is the current. If you understand all of these signals, go for it. Anyway, it produces these curves, and I, of course, was grateful that my co-author, my collaborator on this, understood all of this and could convince me that these are genuine signals, picking up dopamine at this applied voltage and serotonin at this applied voltage, and then here you see the 0.1 nanoamps per second on the y-axis scale. In these two cases, what we're looking at is before cocaine and after cocaine. So cocaine obviously increases dopamine and serotonin significantly. Note the scale on the y-axis has changed. About the same height is equal to 0.2. Excuse me. The nanoamp per second here is the ex vivo calibration of the instrument. This is the in vivo, sorry about that, and therefore it is in picoamps in this case. And of course you can calculate the area under the curve for the dopamine and the serotonin and get a concentration of the transmitter contained within the interstitial fluids. So it hasn't been removed from the brain at all in this case. So you still have to calibrate the instrument, and the signal generated in solution ex vivo for calibration might be different than what you would get in vivo by, in this case, going from nano to pico, from negative 9 to negative 12, as you know. So this data can be collected. This is just a single sweep in the brain, so it can be accomplished within basically a minute or so, and then you have to wait a minute to 5 minutes in between sampling, but you get this very high temporal resolution that you can correlate with other observation. In this case, it's behavior. So we correlated the serotonin levels in various regions of the brain. This is in the dorsal striatum. The serotonin signal in each of these is exactly the same over a 2-hour period. And the filled circles are different behaviors. In this case, trying to read it's rearings. This is central ambulation, staying in the center of the test cage. This is just general ambulations, so it's high just after putting them into the cage. This is animals that were placed into a cage and allowed to habituate to the cage. So within 1 hour, you can see their activity levels drop down to basal levels as they are just found a nice corner to sit in and relax. So when their activities are high, the serotonin levels are the highest, and then it gradually drops off as they become inactive. And the dorsal striatum is a motor area, so you would expect that type of a correlation with the motor behavior. And then the final one we want to consider is the push-pull perfusion. In this case, it's the same thing, except you're actually pushing out into the interstitial fluid compartment itself. A little pocket is created there for the exchange of fluids. So there's an inner cannula that's the push, and the outer cannula is the pull. And you actually have a pump pushing, in this case for 2 separate cannula that are implanted, and a pump that is pulling. So you've got the push, pull out to the fraction collector for each of the cannula, in this case a little rabbit in its octagon there. And it basically gives you an ex vivo sample similar to the microdialysis, but you're causing a tremendous change in the extracellular fluid compartment that is considered a serious limitation for this. This approach had to be used for peptides, larger peptides especially, before the microdialysis tubing became available to collect those types of molecules. It is much more affordable. You can generate these yourself out of steel tubing, so it still is practical, but there are the limitations in interpretation. Again, you have this temporal period. You have to collect samples for 5 or 10 minutes, 20 minutes maybe, before you can detect any of your analytes. So the temporal resolution is very low. Let's just take a few more minutes to talk about this abstract and see the utilization of the push-pull when it was implanted in the anterior pituitary to look at GnRH secretion for a peptide. So using this push-pull perfusion, they measured concentrations of the GnRH in the extracellular fluid at the anterior pituitary in male rats. So they were also basically picking up the hypothalamic releasing hormones in the fluid that they extracted from the pituitary gland. They measured GnRH release at specific times before and after castration in response to acute administration and in response to acute administration of an agonist for the glutamate receptor, the NMDA. After castration, 7 days, the mean GnRH levels were substantially increased, 4.3-fold. So we see the effect of negative feedback compared with removal of the negative feedback, compared with intact controls, so the 0.9 plus or minus the standard error of the mean, 0.16 versus 0.22 plus or minus 0.08 picograms per 10 minutes respectively. So this is about 4.3 times less than what this is. This is obviously in the castrated, and this is from the controls. Significant difference at the p less than 0.05 level. So this was due to an increase in both the frequency and the amplitude of the GnRH pulses. So remember that the pulsatility of GnRH is extremely important, not just the frequency, but the amplitude, the amount released within any one pulse. The testosterone administration partially reduced the GnRH, bringing it down to 0.6 plus or minus 0.1 picograms per 10 minutes, so it's somewhere in between these two values. The NMDA induced a rapid increase in plasma LH in both the intact and the castrated animals, and it increased the GnRH concentrations in the perfusion samples at a probability statistically significant level of p less than 0.05. There was no change in the LH release induced by 2 doses of injected GnRH 5 and 25 nanograms per 100 gram body weight 2 days after castration, but 6 days after castration, the responses to both doses was significantly increased. So this means that the anterior pituitary cells were being bombarded with so much GnRH, pulsatile-like, that additional GnRH could produce no additional effect. So initially there was probably a downregulation of the GnRH receptors, and it recovered after 6 days, so that they could respond to extracellular additional exogenous GnRH. So these results demonstrate that GnRH release in the male is acutely increased by NMDA. So we suspect glutamate synapses are the gas pedal on the GnRH neurons, and is chronically increased after orchidectomy, which is what we'd predict. Increased pituitary sensitivity to GnRH also contributes to the hypersecretion of LH after castration, particularly at longer times after removal of testosterone-negative feedback. So ultimately, by 6 days, the removal of the testosterone would lead to an increased expression of the GnRH receptor gene, and we see the increased sensitivity to the GnRH. And then this final figure is showing the neuropeptide release in the paraventricular nucleus after injecting colchicine into the ventromedial nucleus. So this will dissociate the microtubules and block the transport and release. Here notice 3-hour perfusion period with about 18 perfusion samples, so roughly 30-minute sample collection time periods to produce a single fraction. So this is an example of using the push-pull perfusion to look at a neuropeptide Y, in the paraventricular nucleus of the hypothalamus. And by injecting the colchicine into the ventromedial nucleus, they were testing if that might be the nucleus of origin of the axons containing neuropeptide Y within the hypothalamic paraventricular nucleus. So here we see the levels of the neuropeptide Y in the colchicine-treated animal over in each fraction over the sample time period of 3 hours. And in the control saline-injected animal, it's obviously higher over that entire period. The data that can be easily analyzed with a student t-test is when you average these levels over that 3-hour time period, and you can see picograms per 180 minutes shown here, and it's about 110 in the saline-treated animals and barely 25 in the colchicine-treated animals. So we've seen a couple of examples of the utility of these methods to study neuropeptides and neurotransmitters, and I'll stop there and thank you for your time and attention, and hope to see you again soon. Bye-bye.